Welcome to section 1. In this section, we will look at the history of antibiotics, how they work, and their impact on medical practice. Often there is confusion on the use of the terms antibiotic and antimicrobial. The term antimicrobials is an umbrella term referring to several kinds of medicines, including antibiotics, antivirals, antifungals, and antiparasitics. Antibiotics prevent or treat infections caused by bacteria. Antivirals focus on viruses, antifungals focus on fungi, whereas antiparasitics are for parasites. The focus of this module is antibiotics and antibiotic resistance. Microorganisms or microbes have threatened humanity with infection throughout history. The pre antibiotic era, which refers to the time before antibiotics were ever developed, minor infections such as small cuts caused severe illness and deaths. Microbes are not only dangerous but also spread infection at a very rapid rate, resulting in epidemics or even regional and global pandemics. In 1928, the first true antibiotic was discovered accidentally. Alexander Fleming noticed that penicillin produced by the fungus Penicillium killed disease causing bacteria in the lab. However, only in the 1940s did large scale production of penicillin begin. Since then, many other antibiotics have been identified and developed. In 1909, Paul Ehrlich, a German physician, discovered arsphenamine, marking it as the first effective treatment for syphilis and the first modern antibacterial. This discovery laid the groundwork for the development of drugs targeting bacterial infections. In 1928, Alexander Fleming, a Scottish bacteriologist, accidentally discovered penicillin, the first true antibiotic. However, it took more than a decade for penicillin to be refined, produced, and introduced as a treatment for bacterial infections. In 1930s, Prontosil, the first commercially available antibacterial, was developed by Gerhard Domag, a German biochemist. This sulfonamide marked a significant advancement in the treatment of bacterial infections. In 1945, penicillin was introduced on a large scale as a treatment for bacterial infections, significantly impacting medical practice and patient outcomes during and after World War II. The period from 1940 to 1962 is considered the golden era of antibiotic discovery. During these years, most of the antibiotic classes that are still in use today were discovered and introduced to the market. This era was characterized by rapid advancements in microbiology and chemistry, leading to the discovery of drugs that could treat a wide range of bacterial infections. Despite the previous decades of rapid innovation, the 1970s to 2000 saw a significant slowdown in the discovery of new classes of antibiotics. The reasons for this decline are multifaceted, including the scientific challenges of finding new antibiotics, reduced investment in antibiotic research, and the increasing complexity of regulatory requirements. Generally, antibiotics work by disrupting essential processes or structures in the bacterial cell. Depending on these effects, an antibiotic is said to be bactericidal or bacteriostatic. This either kills the bacterium or slows down bacterial growth. Antibiotics can also be narrow or broad spectrum depending on the range of bacterial types they affect. Narrow spectrum means the antibiotics are active against a selected group of bacteria and therefore treat specific infections. Broad spectrum refers to antibiotics that are active against a wider number of bacterial types and may therefore be used to treat a greater variety of bacterial infections. It is important to understand that antibiotics have different bacterial targets or act on the same target but in different places. In principle, there are three main antibiotic targets in bacteria: the cell wall, or membranes that surrounds the bacterial cell, the machineries that make the nucleic acids, DNA and RNA, the machinery that produce proteins, the ribosome and associated proteins. Antibiotics generally act on pathogens to minimize unwanted side effects. For an antibiotic to be effective, it has to act selectively on processes that are unique to the target pathogen. It has to penetrate human tissue to reach the site of infection, should be able to act at a concentration which is not toxic to the patient. Antibiotics can be classified in several overlapping ways, depending on how they work. This can be based on 1. The effect of the antibiotic on the bacteria, and this can be bacteriostatic, for example, tetracyclines, or bactericidal, such as cephalosporins. 2. The range of coverage, which can be narrow or broad. 3. The mechanism of action on the bacteria. And lastly, the chemical structure, which can be based on antibacterial agents. Understanding antibiotic classes helps us select the appropriate drugs that are more likely to be effective against an infection. It is no secret that antibiotics have revolutionized medicine by enabling the treatment of infectious diseases, prophylaxis of infections, safe surgeries, complicated interventions like brain surgeries, critical care, intensive care, renal replacement therapy, among others. Antibiotics have also led to an overall reduction in mortality and morbidity from infectious diseases, as well as an increase in food production, reduced loss of production from diseases.
And now, a page from our Sunday morning almanac. September 28th, 1928, 86 years ago today, a day which changed medicine completely by accident. It happened in the laboratory of Alexander Fleming, a Scottish scientist who had returned from his summer vacation to discover a strange occurrence in a petri dish. I certainly didn't plan to revolutionize all medicine, Fleming later said, but I guess that's exactly what I did. What he found in that petri dish was a mold that had drifted through the air from another lab in which had stopped the spread of a bacteria culture Fleming had been growing, dead in its tracks. Though he didn't have the resources to advance his discovery, other researchers did. And by the 1940s, they had created an antibiotic from the same type of mold, an antibiotic they called penicillin.
This amazing drug, perhaps the medical discovery of the war.
Penicillin saved countless Allied lives during World War II and won Fleming a knighthood in 1944 and in 1945 a Nobel Prize. Sir Alexander Fleming died a popular hero in 1955.
Through mass production methods, America is continually increasing its output of penicillin.
And though millions have benefited from its discovery over the years, penicillin and other antibiotics are now losing much of their effectiveness in the face of growing bacterial resistance. All the more reason for today's medical researchers to watch out for peculiar developments in their petri dishes.
This Tiny Teach is going to discuss the targets of antibacterial drugs. The first target is the bacterial cell wall. These antibacterial drugs block the synthesis of peptidoglycan, resulting in the lysis of the bacterium due to changes in osmotic pressure. The second target is disrupting or disorganizing membranes. All cells, both prokaryotic and eukaryotic, have a cell membrane. Because of this, drugs that target membranes are not used often because they're toxic to both human cells as well as bacterial cells. Polymyxins are used rarely due to toxicity to kidneys, but are used to treat some drug resistant infections. The third target is nucleic acid synthesis. At first, it would appear that DNA or RNA synthesis would be a bad choice since prokaryotes and eukaryotes both have DNA and RNA. However, there are differences that make this a good target. The bacterial DNA polymerase is different from the eukaryotic DNA polymerase, and the bacterial DNA gyrase is different from the eukaryotic DNA gyrase. Both of these enzymes, therefore, are a good drug target. The fourth target is to block protein synthesis. Prokaryotes have 70S ribosomes and eukaryotes have 80S ribosomes. The fact that the ribosomes are different sizes make them a good antibacterial drug target. Bacteria need to produce folic acid and use that folic acid to produce DNA and RNA. The final target is metabolic pathways. Here, the antibacterial drug acts as a competitive inhibitor and occupies the active site of the enzyme so the substrate can't bind, thereby inhibiting metabolism. In this case, stopping the production of folic acid, which is needed to produce DNA and RNA. This does not harm humans because we get folic acid from our food. We do not produce it in the same way that bacteria do.